Hello and welcome to Lab 7B. My name is Sam and I will be your TA for this session. Today we'll be covering the second session of conjugation as well as the second session of genetic regulation. In this session of conjugation, we'll be plating out the cells from our mating plate that we created earlier in the week to determine if either the plasmids containing either glide A or glide B have restored motility to our gliding motility mutant. We'll be able to see the gliding motility restoration as the bacteria will spread from where they were streaked on the plate. The procedure for this experiment will go as follows. We will be scraping the cells off our mating plate with a sterile applicator and then box streaking them onto two different PY2 plates, one with erythromycin and then one PY2 plate without the antibiotic. Now, if you're unsure what a box streak is, you can turn to page 44 in your lab guide or watch the demo video for this experiment, and it will be explained in both of those locations. So, as I said, we'll be streaking them onto two different PY2 plates, one with erythromycin and one without erythromycin. Now, this is important because erythromycin will be selecting for F. Johnsoniae, because only F. Johnsoniae will be able to use the antibiotic resistance gene that is located in the plasmid. So therefore, it will kill off any E. coli that may have also been growing in culture. So we'll be doing this for each of our conjugation plates, which we have 4, so that brings our total to 8 total plates. We'll be incubating these at room temperature in a sealed container to make sure they do not dry out. Onto genetic regulation. So the purpose of this lab is to determine what mutations have occurred in the different strains of E. coli. So 2 of the strains are going to be mutated, and 1 is going to be wild type. We're going to be determining this by doing a beta-galactosidase assay. For this session, there will be 3 different materials that we will be using, and I'm going to explain what they do so that way when I mention them in procedure, it will make more sense why we are adding them when we add them. So the first is toluene. What toluene does is it stops enzyme synthesis and kind of creates a freeze frame of what is happening in the cell at that certain time. And it also permeabilizes the cytoplasmic membrane to allow ONPG, our next material, to enter into the cytoplasm. So what ONPG is it's a substrate of beta-galactosidase, which when cleaved by beta-galactosidase, it yields a yellow compound, ONP. So the more yellow the culture turns after adding ONPG, the more beta-galactosidase is present in the cell. And then finally, our last material is potassium carbonate. Potassium carbonate will stop all reactions so we can observe and record the differences between the tubes that we have added ONPG and toluene to. The procedure goes as follows. We will mix to resuspend any of the sediments from our culture tubes we created in the first session of genetic regulation. Then, in a fume hood, we will add 10 drops of toluene. We then mix and incubate for 15 minutes at 37 degrees Celsius. This allows the toluene to permeabilize the cytoplasmic membrane and stop all enzyme synthesis. After that, we will add 1 ml of 1% ONPG to each tube. We will mix and then incubate for another 15 minutes at 37 degrees Celsius, or until at least one tube turns yellow. As I had said before, ONPG is a substrate of beta-galactosidase that will yield a yellow compound, ONP, when it is cleaved. After the second 15-minute incubation in a 37-degree water bath, we will be adding 2 ml of 2% potassium carbonate. This will stop the reaction and give us a chance to observe the tubes that we have prepared. We'll be judging the intensity of the yellow color compared to control. We'll record a negative sign for no color and record a plus sign for the faintest color. And then the scale goes all the way up to 4 plus marks. Now, I will be including some examples in the following slides to give you an idea of the varying intensities you can see in this experiment. Here are some examples of the varying intensities of yellow you can see with the tubes. The discussion of results for this lab will be a little bit different than previous labs. Each of you will be receiving a picture of a set of tubes from your TAs. You will then score the tubes and post your results to the discussion board by Sunday for your attendance performance points for Lab 7B. There are several things you should be including on your discussion post. First of all, the strain. This will be either 012, W575, or ML308225. You will then want to include the scores for each color cap, red, blue, and silver. Make sure you list which result corresponds with which color cap. So in today's lab, we went through the second session of conjugation. The lab report for this will be due in a couple of weeks. We also went through genetic regulation second session, and there will be a lab report for this, and you should be sure to include the questions from the supplement. Next week, we will be finishing conjugation and phototrophic bacteria, as well as starting fermentation and determination of bacteriophage titer.